So hello everyone and good morning. Day 2 of the single cell RNA-seq course, and I'm very happy to see all of you guys and meet all of you virtually. My name is Nikolay Oskolkov. I'm a Salaf Lab bioinformatician, and I'm going to talk about dimensionality reduction and integration today, single cell data integration. And just to tell you a few words about myself. So I'm originally coming from theoretical physics, and then I switched to biomedicine, did lots of medical genetics at Lund University. And since 2016, I've been working at NBIS Lab. During this time period, I was involved in different projects, varying from basically single cell to ancient DNA. And today I will be sharing some knowledge, what I learned working on a few single cell projects in terms of what I learned about dimensionality reduction. And just to start with, I wanted to remind you, I don't know, maybe it's not everyone realizes it, that dimensionality reduction is not only a visualization tool. I think it's important to keep in mind that many of us think about dimensionality reduction as a way to get nice UMAP pictures of cells. But please remember that the original goal of dimensionality reduction is actually to reduce dimensions. So so this is what we typically understand by dimensionality reduction. For example, this is a benchmark, machine learning benchmark called MNIST. It's a data set of black and white images, of handwritten digits. And people typically use this data set as a proxy because it reminds these clusters that you get with t-SNE and UMAP. They remind about single cell clusters, so it's quite popular, or I would say gold standard. Whatever new machine learning dimensionality reduction method pops up, they typically test this method on this MNIST data set. And this is how a PCA plot looks like on the left-hand side, and on the right-hand side, this beautiful t-SNE. And this is how we typically, many of us tend to think about dimensionality reduction. It's a way kind of to visualize your data, to explore your data, and that's certainly true. It's certainly a way to visualize as well. But I personally tend to think about visualization as a byproduct of dimensionality reduction. And the major goal is actually reducing dimensions. It turns out that lots of biological data are high dimensional, meaning that we are simultaneously working with many thousands of genes or many millions of genetic variants or CpG sites. And the number of variables, biological variables we analyze, it sets the dimensionality of our data. And typically, for good, robust statistical analysis, you would like to have as few variables as possible and as many statistical observations as possible. So we typically have much fewer statistical observations than number of dimensions, number of genes. So maybe we are working with 20,000 genes or millions of germline genetic variants, but we don't always have sample sizes of many thousands or many millions. So it's quite rare still, and that's why you typically can run into problems in statistical analysis, various problems, various pitfalls. So I'm not sort of going into the bushes here. I'm not explaining what exact pitfalls, but I highly recommend you to check the literature on the curse of dimensionality. So the major obstacle here is there is a common statistical pitfall called the curse of dimensionality, and that's literally problems with convergence of equations when the number of dimensions exceeds the number of statistical observations. If you are interested in this topic and want to, understand deeper where exactly the danger comes from, why this, high-dimensional biological data can be difficult to analyze, and why we need dimensionality reduction, actually. So I highly recommend you to check this series of Nature Methods papers from Naomi Altman and colleagues, where they explain in details basically how you see this curse of dimensionality manifested in your data. And one way to overcome the curse of dimensionality is actually to do dimensionality reduction. So that's a motivation why we are doing dimensionality reduction. So not only to visualize our data, but also to overcome the curse of dimensionality. So next, there are different dimensionality reduction techniques. They can be linear and non-linear. Generally, can be divided roughly in these two categories. Here I'm visualizing the same data set. It's a cancer-associated fibroblast from one of my papers from 2018, and it's the same data set visualized with multiple linear dimension reduction techniques and non-linear dimension reduction techniques. So just to give you a flavor that there is a variety of different dimension reduction techniques. So we typically use not all of them, but just 1, 2, 3 of them. For certain reasons, so it has something to do with the peculiarities of our data. But in principle, there are many more dimensionality reduction techniques, and the linear dimension reduction techniques on this slide, they typically have something to do with matrix factorization. So they have something to do with linear algebra. So we are multiplying matrices here. While non-linear dimensionality reduction techniques, they are quite different. They usually have something to do with graphs. So we build graphs and then trying to project this graph on low dimensional space. So they're technically working in a very, very different way. That's why they look quite differently. So I'll start with linear dimensionality reduction techniques, such as MDS and PCA, what people typically use in life science and computational biology by default in many cases. And this is just an illustration of what essentially linear dimensionality reduction does. So you literally represent your original data matrix X as a product of 2 or 3 other matrices. So on the right-hand side here, that's an approximation. So you approximate your original data X with a product of some so far mysterious matrices, and then you're trying to minimize the difference between your original data and your approximation through this cost function, which can be just a squared difference between your original data and your approximation. And like graphically, visually, it looks like this. So you approximate your matrix X with dimensions n by p. You approximate it by product of U and V matrices, and the matrix U, it has dimensions n by d, and the Lodians, the second matrix, V matrix, it has dimensions d by p. This red matrix, the U matrix, this is what is called embeddance or low-dimensional data representation. So that's actually how you replace your original data. You replace it with this essentially red matrix, and you say that from now on I'm working with this red matrix because that's my new data representation that I'm going to use for downstream analysis, and it has much lower dimensionality than my original matrix. So it turns out that this dimension D, the newly introduced dimension, this product of these two matrices, if you're familiar with matrix multiplication, this product of the two matrices, it's insensitive to exact value of D. So this product is mute. This dimension D is called mute dimension. So it basically disappears once you once you've done the matrix multiplication. So the product will be insensitive to the exact value of D. And here it opens sort of lots of opportunities for you. This D dimension can be much smaller, dramatically smaller than P, the original dimensionality of our data. So by P, I mean the number of genes, for example. So you can go from maybe 20,000 protein coding genes to just 2 or 3 dimensional data. So you replace your original 20,000 dimensional data by 2 or 3 or 5 or 10 dimensional data, but it's a dramatic dimensionality reduction. So I hope you see the message why it's called dimensionality reduction, because we replace our original matrix N by P by new matrix N by D, where D is smaller than P. And then the next question is how to select this D. What D is good? Is my 2D UMAP good enough to represent my original data, or should I consider multiple components? So that's not an easy question. And talking about linear dimensional reduction techniques, I would recommend you to use this approach, which is called Tracy-Widom approach. It's a way to select a good number of principal components. So I'm not kind of explaining, PCA in details algorithmically, just to concentrate on more important things. But here, so what I'm showing, it's strictly speaking, it's how MDS algorithmically works. PCA does not do exactly algorithmically this operation, but it's conceptually it does something very, very similar. And a way to estimate the number of this what is called latent variables and principal components. Those are latent variables. Those are new pseudo genes, pseudo representations of your original variables. I would recommend you to use the Tracy-Widom approach to estimate a good number of principal components to keep for your downstream analysis. And the way it works is that, for example, once you computed PCA, I'm quite sure that I don't have to explain that one of the outputs of PCA is this so-called scree plot or explained variance plot. So basically here, what I'm showing here on the right top corner is how much variance in your original data is explained by each principal component. Along the x-axis, those are principal components, 1, 2, 3, 4, 5, etc. So if you produce, and on the left-hand side, here is a PCA plot of MNIST, the collection of images of handwritten digits. So basically, if you want now to estimate how many principal components I want to keep, you can use some common sense and say, well, I believe that principal components starting from maybe 20 or 40, this tail of this distribution of explained variance, it explains very little of my variance. It's below 1% of variation. So maybe I should cut this tail somewhere here at around maybe 40 or 60. And that's one way, what is called elbow approach, when people just use their intuition and introduce some little bit arbitrary threshold and just drop principal components that explain very little variation. There is another more sort of maybe a little bit more scientific approach. When you compute what kind of explained variance scree plot you would expect from shuffled data matrix. So you take your original data matrix, your MNIST, pixel intensities or gene expression matrix for single cell RNA-seq, and you just perturb it. You shuffle it, you randomize it. so that elements of the matrix, they jump on some random positions within the same matrix. You just shuffle rows, shuffle columns, and you compute PCA on this shuffled matrix again. And you do it many, many times, and you average across this explained variance on a shuffled data matrix. And what I'm plotting here, the blue curve is exactly this observed, what I call observed explained variance on the MNIST data set. And the red curve, this is what you get, a variance explained on a shuffled matrix. And at a certain point, this blue and the red curve, they meet each other, maybe at around 60. Visually, it looks like this. And this is basically what sort of sets a noise zone. So you can say that principal components starting from PC60, they explained variance below, the noise level. Below what can be explained by chance, by what can be explained on a shuffled matrix. And you can even do a statistical test, compute a p-value, and this sudden jump around 60. This what can give you a good number of principal components to keep for your downstream analysis. So I think the idea, basically what is called in literature trace rhythm, but in Surat, it's called Jackstraw. It's not exactly this kind of procedure, but it's also based on randomization. So you basically shuffle your data in order to estimate what principal components to keep, and run clustering, for example, on those principal components. So PCA, just a few words about PCA. Maybe it's a slightly less popular technique in single cell analysis, or maybe I'm wrong, but it's just my impression. But if you look around for genetic analysis, for example, it's been a gold standard. People noticed a long time ago, in 2008, starting from this John November paper, that samples, if you compute a PCA on genetic variants from individuals across Europe, you can basically reproduce the map of Europe. So that there is some sort of correlation between genetic distances and geographical distances. At least that was the hypothesis to test. And it looks like, at least from this paper, it looks like there was some correlation. Well, still, this PCA approach for genetic distance and correlation with geographical distances, it's still criticized from time to time. And especially there is one known pitfall about PCA, is what is called a pitfall of uneven sampling. It turns out that PCA is quite sensitive to the reference clusters. They should be more or less equal in size. So what I'm showing here, it's 1,000 genomes. So this is not so far gene expression. So it's just a demonstration of this uneven sampling problem for PCA, which can be, of course, also replicated for single cell gene expression data, because it's like a common thing, natural for PCA. So here I'm visualizing 1,000 genome data. It's human populations from Africa, Europe, and Asia, and there is some query population, Mexican, what self-identified Mexicans. And then if you ask a question whether this query population should cluster closer to one of those three reference populations, let's call them African, Asian, and European, then this clustering of the query population with respect to the reference clusters can be affected very much by whether one of those reference clusters is a little bit smaller or a little bit bigger than the other two. For example, here on the PCA on the left-hand side, I downsample Europeans. So originally, all three reference clusters, they had 500 individuals, but now I downsampled Europeans, I think, down to 100. And the Mexican cluster, it shifted towards European, but the situation is completely different. So the Mexican cluster kind of moves towards Asians if I don't sample Asians. So basically, this query, the position of the query population, in a funny way, it can be shifted between two reference clusters, and you come to dramatically different conclusions. Because in population genetics, for example, people want to know, they want to know whether my query population, what is the genetic relationship of my query population with respect to other world populations. And so please be careful if you are looking at PCA maps. Please make sure that if you're projecting a certain population on an atlas, and this is what is quite relevant to single cell atlases as well. So if you project a query population on an atlas, please make sure that the reference populations are sampled evenly, what is called even sampling. Otherwise, and if you are using PCA for this purpose, otherwise there can be this, misinterpretations that the query population can end up wherever, so it gets attracted to undersampled cluster, reference cluster.
When we get a tissue, for example, some cells tend to be more represented in that tissue, and we still want to use PCA. Is it viable to kind of subsample all of the different cell types that we have identified to like a specific number and then create a PCA with that and then reintroduce them later, the other cells that we've removed for the PCA for downstream analysis?
Yeah, exactly. So if you indeed, so exactly what you said. So if you want to, project cell population to an already established atlas, so please at least keep in mind this effect that your query cell population can end up, and if if the position is actually important for you, like for functional interpretation, maybe you want to say that my query population is closer to that cluster. That's why it's functionally closer. These cells are functionally closer.
Yeah.
To that cluster. So if this type of interpretation is your kind of research question, and you are using PCA for this projection, so please keep in mind the sensitivity of PCA with respect to this uneven sampling. And it comes from the fact that if you check sort of the equations, how PCA is computed, so it turns out that you have to compute a variance covariance matrix, and there is a denominator, a common denominator for all cell populations or world populations, n. When you compute, so basically your variance covariance matrix is divided by n, by the total number of individuals, and it assumes it implicitly assumes that all your populations are of the same size.
I see.
Which is not true. So basically, the way you compute PCA, so PCA is unsupervised learning. So it doesn't know anything about populations. So it doesn't know which cluster big, which cluster is small. So it assumes that all of them are more or less equal, of equal size. And that's why when you compute, variance covariance matrix, this kind of equal representation of all clusters is already coded in the equation. That's why this effect becomes visible. So please just keep it in mind. So there are sort of there are workarounds here. So you can renormalize your variance covariance matrix, like computationally you can compute it in a better way. Or as you said, for example, you can downsample your reference clusters. So, but also had a suggestion.
No, not a suggestion. I have a question. So here you're only visualizing the first two components. If you would add in more components, would this effect not be as strong then?
Yeah, absolutely. Yes, I'll come to this. That
okay, sorry.
two principal components are absolutely not a good representation of your original data. But typically, what people in the pop gen at least people don't look at more than two or three components because I mean there is a good reason for this. I mean we can't visualize five principal components in a good way. That's why, yes. So an additional problem here is that you are looking at two principal components only. Yes. So, but I was going to come to this in one slide. So basically, in single cell analysis, I think PCA was never popular because people immediately realized that all cell populations, they clump together on a PCA plot if you're looking at two principal components. And UMAP was, or TSNE was kind of much better. So please compare these pictures on the left and on the right hand side. On the left hand side, basically you can't resolve these 12 PBMC cell populations from Surat benchmark data set. And on the right hand side, it's quite visible. And the solution here, so basically what we see here is that there is nothing wrong with PCA. So it turns out that two principal components are not informative enough for our purposes. And so you either should consider more principal components in order to get a better picture. So for maybe 5 to 10 dimensional space, all these 12 cell populations would be resolvable, but not in 2D PCA. But on the other hand, UMAP and TSNE, they have this magic to combine multiple principal components into just 2D. So for UMAP and TSNE, 2D map can be more informative, than 2D PCA plot. And you can see it also here. So what I'm showing here is basically it's a simulated data matrix with 3 populations. So I'm simulating, you can say, gene expression matrix, and there are some marker genes that are expressed in some cell population and not expressed anywhere else. So so basically I simulate 3 well-defined populations, and if I do a PCA or TSNE on this simulated data matrix, then both of them tell me the same story. Basically, I see the 3 clusters, 3 populations that I sort of ground truths that I simulated. But the situation becomes very different if you increase the complexity of the data. When you increase, for example, the number of populations. So if you look at 10 simulated populations, then TSNE can resolve very well all 10 of them. And this is not the case for PCA. On the PCA plot, we can see that we can see 4 basically clouds, so there is no. Indeed, it turns out that PCA two principal components are not good enough for visualizing your data when the data is complex enough. When the data complexity, such as the number of populations in the data set, grows. And in PopGen, people typically work with a few human population. That's why PCA is kind of good enough for PopGen analysis. While for single cell, we are used to, I think, working with 10 or 50 populations if we are talking about big atlases. So that's why TSNE, well, that's one explanation why TSNE can be more informative than PCA for single cell data. And also, just to reiterate it, PCA works what is called on linear data. And here I'm just showing one simulated ground truth. So I'm simulating kind of cell populations which look like continents. And since I simulate them in a world map fashion, so since I have some feeling about distances between the continents, I can now apply PCA, TSNE, and UMAP and see how much they can perturb the distances between the continents. And PCA does a great job, so it basically reproduces the original ground truth, while TSNE and UMAP they kind of distort. They distort the distances between the continents, and you can say, well, why do we need them? at all. Why do we need this nonlinear dimensionality reduction techniques such as TSNE and UMAP if PCA does a great job? Well, it does a great job here for this particular data set because it was like a linear sheet, world map, and it becomes very different if you try to, wrap this world map in what is called Swiss roll. So if you introduce one more dimension, so if you go from 2-dimensional world map to 3-dimensional world map, but just you wrap this original world map as a Swiss roll, and one dimension here is actually redundant. So a good dimension reduction technique should reproduce actually the original 2D map. Because the third dimension here kind of doesn't bring any new information. The distances between the continents, they're all coded within 2D map. And this is what PCA does with this situation. So obviously, PCA might fail for some, complex data sets. So here, this is what people commonly refer to nonlinearity when you sort of introduce, this complex structure in your data. Then PCA might not be optimal for resolving the distances between the continents. And TSNE and UMAP, they do a little bit better job, not optimally, not optimal either, but at least you can clearly see all 5 continents. So that's another motivation why we use TSNE. So basically, sometimes we work with such complex data that PCA, 2D PCA might not be good enough for our purpose. And the reason why PCA failed here was because essentially PCA or MDS, they try to preserve distances between all pairs of data points. So they kind of generate this fully connected graph. And they don't prioritize any distances. They say that all distances between my data points are equally important. And that's not the case for, nonlinear dimensionality reduction technique, such as TSNE and UMAP. They actually explicitly set some prioritization. So they say that my local neighbors are more important than my furthest neighbors. So they don't try to preserve all distances, but they preserve distances between nearest neighbors only. So essentially, you can learn this manifold very nicely. If you go if you go from nearest neighbor to nearest neighbor, you can explore and learn this manifold much better with TSNE and UMAP. Yes, and now we come actually to how these nonlinear dimensionality reduction techniques work, and they do essentially a very simple thing. So there are 3 steps. Both TSNE and UMAP, they do 3 steps. Step number 1, they generate so-called high-dimensional graph. So they represent your original data in this strange graph form, where one node is one cell, and the edges, they're computed. They're basically correlations between your samples or cells computed through gene expression values. So these edges, they symbolize how strongly different cells are related or correlated with each other. So it's just an alternative representation of your original gene expression matrix. So and you do it using all genes. That's why it's called high-dimensional graph. And alternatively, people say, well, just to rephrase this graph construction in a more mathematical form, they say we define a set of probabilities of observing data points at a certain distance in high dimensions, and we call this set of probabilities, P sub ij. And so the step number 2, we also create an approximation for this high-dimensional graph. So we construct another graph using different assumptions, but we are trying to kind of create something that we believe approximate this original high-dimensional graph as much as possible. Or in probabilistic form, they say, well, you define another set of probabilities of observing points at certain distances in 2D. In this case, it's a 2D representation. And yeah, so if you're familiar with Bayesian stats and variational Bayes approach, so it's very similar. So if you can't learn, this posterior, original posterior distribution, you create some approximation, and you are using on a step number 3, you are using some cost function, which for the case of t-SNE, it's Kalbi-Leibler divergence, which basically it's a standard way in machine learning to make two sets of probability distributions as similar to each other as possible. You merge this P and Q distribution and try to make these two graphs or these two sets of distributions as similar to each other as possible through optimization of a cost function, which is called Kalbi-Leibler divergence. So that's it. Basically, in hand-waving explanation is like this. Here it's a little bit more mathematics. So actually, the high-dimensional probabilities, they are defined through this Gaussian kernel. It's basically Gaussian exponent, and here in the power of exponent, those are the distances between all pairs of points. And please keep an eye on this sigma squared in the denominator of the exponential power. This is actually what is related to a parameter, hyperparameter, which is called perplexity. And this is actually the parameter that sets, how far data points can see each other on this high-dimensional manifold. I said that using t-SNE or UMAP, you're looking only at the nearest neighbors. And this is what is controlled by this parameter sigma, which kind of sets a window, a screening radius for sort of how far all data points can feel each other or can see each other. This is what is controlled by this sigma squared parameter. And the low-dimensional distributions, they are coded through this student T distribution, what's called student T distribution. So this is a Kalbi-Leibler divergence. It's this logarithmic combination of P and Q distributions. And here, when you minimize this Kalbi-Leibler divergence, you actually, as a result, you get coordinates y for your data points in a low-dimensional space, in this 2D space. By optimization of the Kalbi-Leibler divergence, you actually get the coordinates of data points, y. So this is a parameter that you actually optimize. And here it's kind of a more explicit set of equations that codes the t-SNE. It looks horrible, but actually it's not that difficult. If you kind of spend a few hours, it's actually not that difficult to code t-SNE from scratch, just trying to code these equations. So here, the equation number 2 is actually the equation for perplexity. It's an important parameter, as I said. And it's basically, if you look carefully at this equation, so you will see that this perplexity, it's actually a fixed number for perplexity on the left-hand side. And this is what decides what kind of sigma squared you will get if you perform the summation. And you will this equation 2, it actually optimizes the sigma squared. Yes, and otherwise you get these beautiful pictures, as I said, by so the output are the coordinates in 2D, and it works very well through gradient descent because Kalbi-Leibler is easy to differentiate, relatively easy. So if you can compute the derivative of the cost function, you can run gradient descent. Yes, so now the question is, so what perplexity is good? People are often concerned and say, well, I don't like that it's a little bit arbitrary, maybe perplexity 50 or perplexity 500, well, what should I choose? So people in single cell, they realize that indeed, especially large data sets, they probably need high perplexity. And Lawrence van der Maten, the creator of t-SNE, he was saying on his web page that, loosely speaking, one could say that larger, denser data set requires a larger perplexity, but no clear instruction how much larger. I think if you look at typical implementations of t-SNE and UMAP, these default parameters for t-SNE, for example, the perplexity, default perplexity should be something like 50, and it will not work well if you're working with millions of cells. So clearly you should increase this perplexity, but no good intuition how much you should increase it. And then at some point I was doing a silly experiment when there were just a few data sets available in single cell in the beginning of this single cell era. So I just collected all these data sets, and I was looking at trying to reproduce the original papers, the t-SNE from the original papers, and I learned that the optimal perplexity that kind of reproduced the t-SNE from original papers, it was varying with a number of cells approximately linearly on a log scale, and then this simple simple fit can give you a simple scaling law for perplexity as a function of a number of cells, and this is a square root of the number of cells. So basically, it's just a rule of thumb. It has some, more mathematical justification. So it's not just empirical fact, but actually in graph theory, this rule is kind of quite known. For KNN graphs, the way they select K, optimal K is actually through square root of n. And so if you are not sure what kind of perplexity is appropriate for your particular single cell data set, just take square root of n of your number of cells and you will not be completely off. And this is my recommendation. So next, t-SNE was at some point kind of criticized in literature. There are a few limitations of t-SNE. Indeed, people notice that it's computationally very hard for large data sets. And then there was this obstacle that t-SNE, as I said, it can look only locally. It can preserve distances between nearest neighbors. And this sets a limitation for your interpretation of clusters. For example, t-SNE, it actually guarantees that it can't guarantee. So if, for example, on this t-SNE picture, you see that this pink cluster is visually closer to the orange cluster, it doesn't mean that they're actually functionally more similar. So you can't say that this pink cluster is actually closer to the orange one compared to the red one. So this interpretation is wrong. Because there is this, sigma parameter in the denominator of a Gaussian exponential power. So this comes from the equations, how actually t-SNE is computed. So it can't, in principle, preserve intercluster distances. So if originally your, clusters, I mean, it can't reliably tell you on this 2D map what was their intercluster positions of the original clusters in the original high-dimensional space. So basically, that's one of the important things to keep in mind working with t-SNE and UMAP, that intercluster distances, they're meaningless. And UMAP, it was kind of suggesting that maybe this problem of global distance preservation of t-SNE can be somehow solved through, there were a few major modifications of UMAP compared to t-SNE. For example, the initialization was changed from random in t-SNE, it was random initialization. For UMAP, it was set to be a Laplacian eigenmap, and the cost function was changed from Kalbi-Kleibler to what is called cross entropy, and there were a few other modifications. So, and indeed, there was a promise just basically from this functional form of their cost function. It looks like so here I was plotting the Kalbi-Kleibler for t-SNE on the left versus cross entropy on the right, and x and y, the x and y coordinates here, those are average positions between data points in high dimensional space x and low dimensional space y. And just by looking at the how this cost functions for t-SNE and UMAP behave, it looks like if data points were far apart from each other in high dimensional space, so when x was large, distances in high dimensional space were large, then the points can be at any position basically in low dimensional space, because the profile, the landscape of this Kalbi-Kleibler was basically flat here, meaning that they can end up. So if the points were at large distances in high dimensional space, they can end up at any distance in low dimensional space. And it was not the case for cross entropy, where you get a penalty here. So if you place your points that were far apart in high dimensional space, if you place them close to each other in low dimensional space, you get this high penalty. So you should place them far apart in low dimensional space. So that's a good motivation, I think, behind using this cross entropy as a cost function for UMAP. Unfortunately, still the sigma parameter in the so this is how high dimensional probabilities looked like for UMAP. This sigma parameter, it still kills just a principal, possibility for global distance preservation. As long as this parameter exists, you can't say that UMAP can preserve a global distance. Maybe you can say that UMAP, and this is what Leland McKeans, basically the author of UMAP, says, you can only say that UMAP probably preserves more of a global structure compared to t-SNE. So these interpretations of, what is the intercluster distances here, they're a little bit more reliable for UMAP, but there's still not much trust should be put on both t-SNE and UMAP when interpreting intercluster distances. Okay, so now there was a so t-SNE people basically at some point there was a claim in literature that large perplexities they kind of improve global structure preservation, and indeed looking again at this benchmark data set, the world map, it looks like if you increase the perplexity, so you start with quite a perturbed world map at low perplexities, and eventually you come to almost perfect preservation of all their intercontinental distances at very large perplexity. And this was kind of suggested as one way to make t-SNE and also UMAP to preserve more of a global structure. You just increase perplexity and then you're fine. You can be more accurate with interpreting intercluster distances. This was what I heard from the t-SNE developers. But then, unfortunately, there is another complication here. When you increase perplexity, it turns out that the gradient of Kalbi-Kleibler divergence, it actually disappears. So if you check, so this is an equation for a gradient descent. It means you update your coordinates, low dimensional coordinates y through using the derivative of the Kalbi-Kleibler with this prefactor mu, which is a learning rate. So it's a standard gradient descent optimization rule. This is how t-SNE and UMAP, they actually optimize, do this optimization of Kalbi-Kleibler or cross entropy cost functions. Well, it turns out that this, the gradient of the cost function, it's very sensitive to large perplexity. So the larger is the perplexity, the lower is the gradient. So it's a little bit related to this vanishing gradient problem in neural networks. If you're familiar, so your neural network stops learning at some point because of this gradient disappears. It becomes basically zero, so you don't update your coordinates anymore. So what it leads to, so like this, so the effect that you observe here, why t-SNE actually reproduces the original world map perfectly, is because if you apply initialization with PCA and increase your perplexity up to 2000, like it was here, so you actually get a PCA. You don't get a t-SNE anymore. So t-SNE never starts properly. So you initialize your t-SNE with PCA, and you end up with PCA at the end, because the t-SNE algorithm never starts. That's the explanation why at large perplexity you might get a perfect intercontinental distance preservation, like here on the right bottom plot. So just another few demonstrations that please be careful with large perplexities. So here for this, more complex nonlinear data sets, so we know that PCA doesn't do a good job here. But also t-SNE basically at large perplexity, it gives you pictures more or less identical to PCA, if you use PCA as an initialization, like I used here. And UMAP is a little bit more stable. So basically, UMAP, you cannot knock it down easily with this perplexity parameter, which is called number of nearest neighbors for UMAP. So even if you increase it quite substantially, you will still get meaningful, pictures from UMAP. It's a little bit more stable with respect to perplexity. Yes, I'll skip this because I'm running out of time, unfortunately. So I'll go to autoencoders as a good, promising way for dimensionality reduction. So it's, if you're not familiar, autoencoder, it's a neural network with this butterfly structure. And the idea of autoencoder is to reproduce the values of the input values. You're trying to reproduce the input values on the output layer as much as possible. So you're on your output layer, you're trying to reproduce your input layer. So it sounds silly. Why would you do it? Until you realize that here in the middle there is a what is called bottleneck. It can be only 2 or just a few nodes, and so basically, if you manage to compress your original data down to 2 dimensions and reconstruct it back without losing much information, it means these 2 nodes in the middle, the bottleneck, these two nodes, they learned something fundamental about your data. This is the idea of dimensionality reduction with autoencoder. And I would say autoencoders, they still have some limited applications for single cell in terms of dimensionality reduction. So they do have other advantages, but as a visualization tool, it looks a little bit better than PCA. So this is a PCA picture from PBMC cells that I showed you previously, and this is kind of an autoencoder picture of the same cells. It's a little bit more resolvable, like all these 12 cell populations are a little bit more resolvable, but it's of course not comparable with t-SNE, for example, that gives you much sharper cluster. That's why I would argue that autoencoders are they're limited. They have limited applications for visualization, for dimensionality reduction purposes, although they are good at data pre-processing. If you want to, pre-process your data using some non-linear activation functions, and if you want to pre-process your data for some other machine learning purposes, then probably autoencoders promising. Yeah, so just final words that I wanted to mention. I'll skip this variance explain because unfortunately no time. There was a recently discussion, so I don't know if you guys were following this, that suddenly UMAP was criticized a lot by the community, especially population genomics community. It turns out that after this publication of the All of Us paper in 2024, like many people objected and said that using UMAP was completely wrong. To visualize human populations with UMAP was too far-fetched. And then maybe cell biology people got worried because, I mean, in cell biology, it's a gold standard and people are using UMAP by default. So if PopGen people criticize UMAP, what kind of reasons they have? And indeed, many people in PopGen, such as Jonathan Pritchard, will, posting that it's inappropriate to use UMAP. And then if you're not familiar with pros and cons of UMAP, you're just a user for your single cell project, you might get worried that probably there is something wrong about UMAP and I should not use it for my projects. And Rafael Irizarry, very competent computational biologist, he published a popular post explaining that that UMAP produces artificial clusters, and biologists, please stop putting UMAP plots in your papers, etc., etc. So I inspected this kind of his arguments in very much details, and we had the discussion. So basically, what he claims that UMAP generates this artificial cluster. So he simulates data from multivariate normal distribution, which is all the data points are supposed to be independent. But what you get from UMAP, you get these two artificial clusters, which is of course very worried. But then it turns out that basically, if you check carefully his codes, there was some small error there. So he was computing UMAP on a distance matrix, and he should not compute UMAP on a distance matrix without informing UMAP that you're working with distance matrix. So basically, I wouldn't be worried that much, about these strong statements that UMAP somehow creates artificial clusters. So probably the situation is not as bad. But indeed, for population genomics, this inability of UMAP to correctly address intercluster distances is very relevant. For us in single cell, often we want to, just discover clusters. We don't want to interpret what cluster is closer to another one. While in PopGen analysis, that's essentially the goal of their analysis is to say whether this Puerto Rican population, if it clusters, for example, closer to this cluster, can we say that this Puerto Rican population genetically closer to Great Britain plus Iberian population? Is it true or not? And that's an absolutely not true. So we can't do this kind of interpretation based on UMAP plots. So we should do this interpretation based on PCA plots, because PCA preserves correctly intercluster distances, which is not the case for UMAP. And that's why maybe I would argue that for population genomics, UMAP is indeed not optimal, because simply because it doesn't guarantee that intercluster distances are well preserved. And in single cell, that's not a big deal. If intercluster distances are not well preserved, that's not a big deal. We just want to see the clusters often. While in PopGen, it is a big deal. So we actually want to, exactly, that's the whole point of PopGen analysis, to understand what is the position of the query population with respect to reference cluster. So I think I'm done. Also, please go ahead with your question.
Yeah, just back to the same thing you're saying, that in single cell, we only want to discover the populations, should be true, but it isn't true. A lot of people are still using distances in the UMAP to make assumptions about what's more similar. It's being used for trajectory analysis and so on. So I think it's still an important thing to emphasize that distances in UMAP preserve global relationships better than t-SNE, but it's still not like a ground truth of like how similar things are in single cell as well as in PopGen.
Correct. I agree. So the small difference is that what I can add is that in single cell, I think this fact that UMAP doesn't preserve global distances correctly is well known. I think pretty much everyone heard about it at least. While in PopGen, at some point, it became a discovery. So maybe 5 years ago, I saw first attempts of PopGen people, presenting UMAP plots in conferences and making interpretations, and I was kind of politely suggesting that do you know that UMAP doesn't preserve this global distance? And it was kind of a discovery at that field. So people thought that UMAP is kind of PCA, which is not. Of course, they work algorithmically, they're dramatically different. But people thought naively that UMAP is a kind of PCA just with better clusters, with the sharper, more distinct clusters. And this is, of course, people learned it in a hard way after this All of Us publication. Although technically there is nothing wrong with All of Us, UMAP picture. Like the picture here, I would argue, is correct. There is no here. Sorry, the picture on the right hand side is correct. There is nothing wrong with this picture. Just the interpretation is up to you. If you color, I mean, UMAP is unsupervised. It just it's black and white. It doesn't tell you anything about population. It just gives you a picture, and interpretation might be incorrect. So if you try to put colors on these clusters and interpret, this is where the contradiction comes. Yes, please. There was a question.
Hi, I just wanted to ask you about, this is probably a very naive question. Have you seen if the TSNE clusters and the UMAP clusters are conserved, or do you get completely different results if you compare one set of data from the other?
So you mean if you compare TSNE plot versus UMAP plot, how comparable they are?
Yes.
I would say if you have enough data points, they are very comparable. So all this, counter examples, like many people can criticize, take UMAP and say, well, look how badly UMAP works. Here, for example, like in this Rafael Irizares post, but he used only 100 data points. Of course, any statistical analysis, not only UMAP, can be very sensitive to small sample size. But if you have, 10,000, good sample size, I would argue that TSNE and UMAP, they're broadly comparable. Although UMAP is kind of, it's a more advanced version of TSNE in terms of number of hyperparameters. So UMAP has many more hyperparameters. So this the number of nearest neighbors, just very famous parameter. But if you actually check UMAP, it has bandwidth and all this learning rate. So many hyperparameters that you actually never use. But if you do use them, you can basically create more advanced plots compared to TSNE to see more nuanced patterns in your data. But otherwise, if you go just for default parameters, TSNE and UMAP, I would say they tell you the same story for good sample size. Okay. That's mine.
I have another question regarding the heterogeneity, because most of the times when we work with tumor cells, they are highly heterogeneous, but they are all tumor cells. When there is a lot of this intra-tumor heterogeneity, like a data set, which is very diverse, what would you explore both TSNE and your UMAP, or would you say TSNE is better in identifying these variances and maintaining these variances, so go for that kind of analysis first to identify specific groups of clusters?
Yeah, I think you should think about TSNE and UMAP as just models suggesting something. So you can, and it's up to you to zoom in or zoom out. You can sometimes you feel like you see clusters or you don't see clusters. So it's up to you kind of to build your hypothesis, to build your intuition by looking at this picture. So it doesn't mean, it's a true representation of your data, but it suggests something that you can further test. So I would think about TSNE and UMAP as something suggesting, making some prediction that you can further validate. So they all need validation. So any statistical model needs a validation. Yeah. So including UMAP. So if it suggests that there are these 1, 2, 3, 4 clusters, so please validate them. Yeah. Just a suggestion, but you often you can't expect UMAP to give you complete truth. So, but I tend to think about them as just a model that gives me some idea where to look further.
Thank you.
You're welcome. Please go ahead.
Yes, thank you, Nikolay. I had a question also related to heterogeneity in tumors. So, in most of the cases, at least for my studies, so we have like different heterogeneous populations, and then we have like very two distinct populations, and then we have all the transitions in between. So, in that case, if we rely on the UMAP, can we say that okay, this heterogeneous, or let's say this transition between the two defined clusters, there will be more closer to one end than the other, or we can't make such a conclusion. We say that just we have a transition and we can retrieve it with trajectory analysis or something. Can we draw any conclusion? That's my question.
Yeah, I think you like a conservative interpretation would be that you see a transition and sort of it's dangerous to do some quantitative to make some quantitative conclusion to see how much transition or how little transition. That would be probably not accurate. But just a fact, yes or no, I think UMAP is okay, at least my feeling. But if you go to to concrete distances, to concrete extracting UMAP components and using them as variables for something for more quantitative analysis, not qualitative, but quantitative analysis, I would be more careful with UMAP.
Can I add to that,
Absolutely.
Nikolay? Because it's also the case, like if you have two trajectories and you want to say that one is closer than the other, for instance, it depends a lot on how many cells did you sample along the two trajectories. How dense they will become in the UMAP also. So basically, the distances you can't really say much about.
Yes. If I just can give you an example, because we have, for example, some of the genes that we know, they are in a transition state. Okay, and then we say we validate in the lab the gene expression of different stem cell markers, and then we can say that this stem cell markers, they tell us they are more epithelial, for example, and some other stem cell markers, which are still stem cell markers, they are more mesenchymal state. And then you don't see such actually clear differentiation in a UMAP. But when we validate in the lab with a qPCR, we see the differences. However, every time we try to propose something, then they say, no, it's not valid. We get criticized about that.
Because it's not there in the UMAP?
Yes.
Oh, that's the reviewers need to come and talk to Nikolay.
You get all of them into just one cluster, and then when we say yes, but these are different, they are still stem cell markers, but we know that some are more specific to this state, some are more or progenitor to this state, but we never get like a consensus on that. So that was my question here.
Yeah, I mean, UMAP can be tuned in many ways. I think maybe you did not explore all possible, all range of hyperparameters because they are kind of, this clustering of dimensionality reduction algorithms. They are just zoom in, zoom out tools, and it depends on you where to stop. So you can say that, all my cells are separate clusters, or all of them together is a single cluster. And so those are two extreme cases. And it's kind of up to your question, where to stop. You apply dimensionality reduction and UMAP from, a situation where all of them are individual clusters, and then they kind of clump together gradually. And at some point you can stop and say, so this is what kind of makes sense for me. This is how I understand my data. And so basically this hyperparameters in clustering algorithms, like resolution in Louvain or perplexity in t-SNE, they can be tuned to get a picture that kind of matches your intuition. It doesn't mean, I mean, there is no kind of perfect right or wrong. You can think about indeed that all your cells are individual clusters, so all of them together is a single cluster. Those are two extreme situations, and but you kind of sometimes you zoom in a little bit and you suddenly see some nuances in your data, some patterns. So it's just, it's a way to kind of learn your data, to get familiar with your data, rather than, giving you a perfect truth.
Yes, thank you. Yes, thank you. That's very interesting. Thank you.
Hey, yes, thank you. Thank you, Nikolay. Very nice explanation. So now it is clear that in UMAP, we don't preserve a distance between clusters. But yesterday, when we were doing the practice, we were talking about doublets. And sometimes doublets, they are breaching two populations in a UMAP. So could this mean that in this case, when we had doublets, that they represent maybe two different cells from two different populations, they actually preserve some sort of distance in the UMAP?
I don't think it's based on UMAP. You do clustering, right? When you detect doublets.
No, actually, in my lecture, I was showing sort of that when you visualize cells in a UMAP, you sometimes get these streaks between two clusters that are the doublets of cells that are like a mixture of those two cell types. So you get sort of, it looks like a trajectory between two clusters, but they're actually doublets. And of course, I mean, we're saying that distances are not, they're not meaningless in UMAP. It's just like you can't really interpret them as true sort of transcriptional changes or trajectory distances or times, but still the layout in the UMAP gives us some interpretation of the data. It's not like it's just throwing things out for fun. We can still interpret some things from the UMAP. So, but maybe you can clarify what you mean with the doublets.
Yeah, no, that was exactly that. So maybe that could be a strategy to reduce, actually, to keep the distances in UMAP. Because yesterday when we kept...
Oh, you mean to keep the doublets, to have these streaks in
Yes.
between?
Yes.
But that's a very artificial way of
Yeah, it is very artificial, but I mean, maybe you artificially introduce them, like you control them, like spike-ins.
Yeah.
And, like some chimeric things. And yeah, of course, it doesn't make any sense because then you have trajectory prediction. But anyway, so yesterday when we got rid of like 20% or 50% mitochondrial, you could see more breaches with the 50%. So then and if you got rid of a lot of mitochondria, then the clusters were more separated and clearly defined. So wonder if having like chimeric doublets very well controlled could be a way of keeping distances in UMAP.
It could potentially, yes.
But I also guess that the streaks that we saw yesterday weren't really like bridging between two clusters, but they were kind of diverging from a main cluster, which could be explained that you have the transcriptomic profile of the original cluster plus some other RNA that makes it slightly more different. So it's, I mean, I don't remember it as being like a streak between two clusters, but rather some cells slightly diverging because they have other RNA profile than the main cluster. But I'm also not 100% sure.
So what you were doing then was doing a different filtering for the percent mitochondria.
Yes, and
so basically, if you include more low quality cells, you get this sort of grouping of cells by just them being low quality.
Yes, you bridge clusters.
But that's a very much an artificial bridge because it's not like the doublet bridge. It's actually low quality cells of different cell types being more similar to each other due to just having low quality than the actual cell type signature.
Yeah, but I mean, when we did the doublet predictions, usually the doublets were in the bridges. Of course, it was just, I agree, very low quality, but
yeah,
just pointing out that could be interesting.
Yeah,